basically, bispecific T-cell engagers are an emerging technology and a very exciting and a successful technology. And the aim of our project was to understand exactly how it works and what causes resistance, what causes patients to not respond, using our single-cell technologies. And we've established a platform to study these questions in different ways, and I'll be discussing two of them. First is an in vitro model, co-culturing T cells and lymphoblasts, T cells and malignant cells. And using an advanced single-cell technology called PEAK-seq, we are able to analyze the crosstalk between these two types of cells and to understand exactly what a T cell experiences when bound to the target cell via the antibody. That was the first part, where we also showed the activation signature. So, and we showed the gene expression related to this process. And then we used primary patient cells in the same model to see how patients differ from healthy individuals. And there we found specific molecules that were missing in the activation and then prevented a response. They basically predicted failure of blinatumomab. So this is the first in vitro part, and also we're working with human patient samples. We currently have a cohort of 10 patients, and we're still enrolling, where we're doing single cell before and after the first treatment of blinatumomab. And then we're examining first the immune composition and its correlation with disease status prior to blinatumomab administration, and also what are the key players in the blinatumomab-mediated response, both in the T cells and among the myeloid cells. So in the T cells, we have surprising findings, basically that despite the fact that all T cells have a CD3 receptor and should be responding to blinatumomab equally, we can see that mostly the memory cells, specifically CD8 memory cells, are the ones that are actually expanded in response to treatment. That's one of the findings. Another finding is the fact that T regulatory cells, which we were afraid that might be also expanded and by that reduce the beneficial effect of blinatumomab, actually haven't changed. And the third finding, well, actually, the third finding relates to myeloid cells. We can see that following treatment, non-classical monocytes, which have been shown to correlate with bad prognosis in ALL, are diminished, and while classical monocytes, and specifically activated monocytes, are enriched. And lastly, there's a question of T-cell dysfunction, both whether the B-ALL causes T-cell dysfunction and specifically whether the treatment of blinatumomab, which is very long, causes T-cell exhaustion. And we are showing that in our patient cohort in bone marrow T cells, we do not see any expression of LAG3, PD1, or TIM3.